Hello, it's Mr. Shrum again, and I am going to go over the nervous system with you, at least the first part about the function, what is a neuron, action potentials, how do neurons actually send information to the brain, synapses, how does a neuron send information to another neuron. And here is the broad overview. We're going to talk about the nervous system, and a great way to think about this is that it is the command center. Every information that you intake from all of your senses, so your visual information, your auditory information, it goes through the nervous system. And the most basic unit of the nervous system is the neuron. These are cells in your body that devotes its function to the nervous system. And there are ways to classify them based on structural capabilities and their function. And the way these things work is as you're taking in information from around you, from the external world, there is a stimulus, and we call those sensory neurons, and there is a response to this stimulus that you and your body internally is doing in response to that stimulus, and those are motor neurons. So I will show you a video from Bozeman Science. Let me share the screen, and he will go over the basic structure of the neuron, the dendrites, the axon, the myelin sheath, and the terminal ends. So let's check that out.
Hi, it's Paul Anderson, and in this video I'm going to go over the basics of the neuron. The neuron is the basic unit in the nervous system. In a simple animal like a nematode worm, they only have 302 neurons, but what they allow them to do is to take information in from their surroundings and respond to that information. If you look at how those 302 neurons are connected, it's very complex. Now, humans are quite a bit bigger, and we have 100 billion neurons in our body. Most of those are centered in the central nervous system, in the brain, and the spinal cord, but the basic unit of our nervous system is the same. It's the neuron. If we look at the neuron anatomy and remove all the other neurons, the center is going to be what's called the cell body. This is a typical cell. It's a metabolizing cell. It's taking in energy. It's using that energy, and so it's going to have a nucleus. It's going to have Golgi apparatus, mitochondria. It's a typical cell, but there are extensions of those cell bodies called the dendrites. Those dendrites are connected to other neurons, and they're taking information in the form of neurotransmitters. In fact, this whole side of the neuron is receiving information. We then have the axon. The axon is where we're transmitting information. It begins at the axon hillock and is going to move to the terminals. Now remember, the terminals, there's a synapse that connects it to other neurons, but the signal is going to move in this direction. We're taking in information from the dendrites, the cell body, and then we can send information to the next neuron. Now, how do we do that is through an action potential, and I have a video that I'll put a link to where I go into the specifics of how that occurs. But certain axons in our body are really, really long, and for that information to travel quickly, we will wrap them in myelin sheaths. It's a Schwann cell that's wrapped around it. The way it works is we put the voltage-gated channels in between that, and it acts to insulate that nerve. It allows the information to travel more readily down that axon. And we generally say if you have an axon that's wrapped in myelin sheath, we call that a nerve fiber inside our body. Now we're taking in information as well. So there's going to be another neuron that's connected to both the cell body and the dendrites of that cell. But there's going to be a gap between these two, and we call that a synapse. It's a connection between neurons. Again, I have another video on the synapse if you want to dig into the specifics, especially of how chemical synapses work. Neurons are classified in two ways. First way is due to their structural classification. So if we take the cell body of a typical neuron, like this multipolar neuron, the way you can figure out what type of a neuron is find the cell body and figure out how many poles are coming off of that cell body. So if we count them, we have a number of different dendrites that are coming off of this neuron, and then we have one axon that's coming off, and so we call that a multipolar neuron. This is the most common neuron that we have in our body. If we look at the next one, we find the cell body again, how many poles do we have coming off of it? We've got the axon coming out and the dendrite coming in, and so we call this a bipolar neuron. Bipolar neurons are very rare. They transmit information from the receptors in our eye, receptors in our nose. If we look at the next one, here's the cell body. You can see there's only one pole coming off of that. It's actually a fused axon and dendrite, and we call that a unipolar neuron. Now, in vertebrates, it's sometimes referred to as the pseudo-unipolar neuron. These are going to be most of the sensory neurons in our body. They're taking information in and transmitting that back into the central nervous system. And then if we look at the next one, if we look at that, you can see there's no axon on it at all. And so we call that an anaxonic neuron. These are found in the brain, and they're not super understood. Now, there are a lot of other structural classifications, but these are the general ones that you're going to find in a typical anatomy class. Now, neurons can also be classified according to their functional classification, those that take in information, stimulus coming in from our environment, and then the response that we have. So if we kind of move that to the central nervous system, we're looking at a cross-section of the spine. So we're taking information in from receptors. So these could be in the skin, for example, touch receptors, heat receptors, pain receptors. And so we have to transmit that information to the central nervous system. So those neurons that bring information in are sometimes referred to as sensory neurons or afferent neurons. You can see this is a unipolar neuron, and it's transmitting that information into the central nervous system. And then we have information going back out. We're sending information out to effectors in our body. It could be muscles, could be glands, and we're going to call those motor neurons. They're moving away from the center. Sometimes we refer to those as efferent neurons as well. Now, there should be connections between those neurons. Those aren't all the neurons we have in our body. And so we also have these connections in the middle, and those are going to be called interneurons. And so that's the neuron. How did you do? Can you remember the different parts of the neuron? Do you remember that these are the dendrites, the cell body? Do you remember what type of a neuron this is? If not, go back and rewind it. But those are neurons. Those are the basics, and I hope that was helpful.
Okay, so if we look at this picture here and do what he says and try to pick out which part is what, do you remember which way the signals flow through the neuron? These are the dendrites. This is the cell, the nucleus here. And this side is intaking information. And that information is flowing this way, sent through the axon towards the terminal ends. And we call this the nerve fiber. I think of this as like a fiber optic cable. If you think of a wire, there is the coil, the metal, and then it is insulated to protect that and speed things along. So that is why that myelin sheath is there, to speed up the action potential chain that is flowing this way towards the terminal end. And then once it's over here, it will give that information to another neuron via the dendrites here. And we should remember the stimulus response as well. Receptors receive information. We also call those afferent. And then the motor neurons, they work via effectors, like effect. It has an effect on the body, and your body responds via the motor neurons. Now, he mentioned action potentials, and we will go to the next video in this series, and I'll probably jump around, but this will help with a lot of the visual learning. I think it's best to look at the sodium pumps, and there's a mnemonic that Mr. Bozeman uses, or Mr. Anderson uses, and it's called Salty Banana, and you'll hear it here. He'll use it, but it's a good way to remember which side has a higher concentration of potassium and which side has a higher concentration of sodium. And this will help explain why some things trigger an action potential and why other things don't, right? And these action potentials work in a chain. And this is how the information is sent along that axon in the neuron. Here we go.
Hi, it's Paul Anderson, and in this video I'm going to talk about the action potential, that characteristic depolarization and repolarization of an excitable cell. I'm going to be talking about neurons, but we could also have action potentials in muscle cells coordinating movement, or in cells of the endocrine system, for example, coordinating the release of insulin. So if we think about a neuron is getting information from its dendrites and cell body. Remember, they're connected to a lot of other neurons, and based on the summation of all of that information in neurotransmitters, it's either going to fire an action potential down the axon or not. And so in this video, we're going to think about three different potentials. The resting potential, that's the resting potential of a neuron at rest. It's not really at rest. We then have a graded potential based on the stimulus that that neuron is going to get. And then we may or may not have an action potential. Now that can lead to synaptic activity on the next neuron. I'm going to go through it in the following order. First, we're going to talk about what a resting potential is and how the sodium-potassium pump and the permeability of different ions in the membrane is contributing to the resting potential. We'll then skip forward to the action potential and the importance of voltage-gated channels. And then we'll come back and talk about how chemically gated channels contribute to either the firing of an action potential or not. And so what is potential? Potential in physics is simply a separation of charge. And so if we think of a battery, a 9-volt battery having potential, that means that there's a separation of the charge within the battery itself. And it also means that we have potential to do work. And so if we're thinking about a cross-section of a neuron, it also has resting potential. What that means is there's a separation of the charges. Now that separation of the charges is going to be across the membrane, just on the surface of the neuron, but we can measure the voltage. It's negative 70 millivolts in a typical human neuron. And so when you're seeing pictures of an action potential like this, on either side of the action potential, we can see that that neuron is at rest. Now, it doesn't mean that it doesn't require energy to do that. And so let's zoom into the surface of that neuron and see what's going on. Now, I've got a little mnemonic that I use with my students to remember the important ions on either side of that membrane. I say the neuron is like a salty banana. Most people remember that a banana has a high amount of potassium on the inside. Doesn't mean that it doesn't have potassium on the outside. There's just a higher percent of potassium on the inside. There are also going to be negatively charged anions. For example, proteins on the inside of the neuron are going to have a negative charge. And then since it's a salty banana, on the outside, we're going to have salt. We're going to have sodium and chloride ions on the outside as well. They're going to be at a higher percentage. We still have them on the inside, but a higher percentage on the outside. Now, why do we have more sodium on the outside, more potassium on the inside? I'll show you in a little bit how the sodium-potassium pump contributes to that. But as you look at this right now, there's still no potential. We don't see a separation in charges. And to really figure out how that works, let's remove a lot of it and just focus on the potassium itself, because it's really the potassium and the permeability of that potassium that's establishing much of that membrane potential. And so we've got a channel on the membrane surface. It's called a leak channel, and it's a leak channel that allows potassium to move out. And so if we think about where is the potassium going to go, there's a chemical gradient from the inside to the outside. In other words, there's a higher concentration of potassium on the inside than on the outside. And since some of it can leak through, it's going to go in that direction. But watch what happens as the potassium moves out. We all of a sudden see a separation in charge. Since this is a positive charge here, it's moving out. We have a separation of that charge, and it's a relatively negative charge on the inside. We also see the arrival of a new gradient. That's the electrical gradient. Since we've got all these positive charges here, they're going to repel each other. They're going to want to move towards the negative charge. And so as more potassium leaves, you can see that electric charge is increasing. And so eventually what could happen is it could start to flow back in. And so we're going to have movement of potassium inside and outside, but we've got an electrochemical gradient outside. Now, why doesn't all the potassium just flow out? It's because these are leak channels. They don't allow much of that potassium to flow out. Now, if we look at a PHET simulation, and I'll put a link to that in the description down below, they've simulated the passing of potassium in and outside of the cell. You can see that we also have another leak channel right here. It's not as busy, but that is a sodium leak channel as well. And so we have an electrochemical gradient of sodium. Remember, there's more sodium on the outside that's coming in. And so we've got these two gradients that are set up. You can see the chloride's not moving. You can see that the proteins are not moving as well, and that's because we don't have a channel for them. Now, if we were to just...
Okay, so I will open up that simulation, take a break from the video to let some of this sink in. But thinking of this as a battery, right? With the potential energy within the battery and the way the potassium and sodium ions reach across the membrane or flow across these leak channels, as he was saying, that creates a difference in these charges, the electrochemical charges. And here we see the sodium, which are the red circles, and the potassium ions. Let's show the charges. So you see how the positive, more negative on the inside. You can also show the concentration, and then the potential chart as well. So if we, I think the concentration, let's get that, it's a little too busy. So we can stimulate the neuron. It has been stimulated, and you see that the potassium ions flow outward, and then the sodiums are influxed. And you can see how he was talking about the membrane potential in millivolts was at the resting potential at -70, and then the excitatory sensory stimulus, right? It's excited up to this potential, and then repolarizes back, and then you have this undershoot, and now you're back to a resting potential. So let's stimulate it again, and let's do slow motion this time. Stimulated right there. Here comes the sodium inside. And that potassium is released. And then if we fast forward, you can see it happen much more quickly. And let's get back to the video.
Just let this go, eventually we would lose that potential because all the sodium would come in. And so we have a sodium potassium pump where we cash in a little bit of ATP, and that's going to move that sodium out, 3 sodium ions for every 2 potassium ions. And so as this is moving and establishing that membrane potential, again, it's the permeability of these 2 ions that's establishing it, that sodium potassium pump is essentially keeping up with the sodium that's flowing in and the potassium that's flowing out. And so that is our resting potential. -70 millivolts is established because we have a separation of those ions on the inside and the outside, and their different electrochemical gradients are establishing that difference in the charge. And so we have this resting potential. Now it requires energy to do that, but what could that resting potential eventually lead to? An action potential. Now let's look at what an action potential looks like. This is that PHET simulation again. And you can see there's a huge amount of motion between the ions on that surface. And as we zoom in, we see our sodium leak channel, we see our potassium leak channel, but we see another couple of channels that are really, really important. And those are the voltage gated channels. How do they work? Well, let's look at the sodium voltage gated channel. So it looks like this. It's got a little polypeptide ball on the bottom. And so when the voltage approaches -55 millivolts, it will open up. And it opens up wide, and that allows a huge amount of sodium to come into the cell. You can see that rush of sodium in that simulation. Eventually, as we move away from that voltage, it's going to be inactivated, and then it's eventually going to close. There's also potassium voltage gated channels as well, and they're going to open when the voltage becomes around 30 millivolts. They'll open wide open. What is that going to do? It only allows the passage of potassium. Potassium is going to move outside of the cell. And so as we look at that action potential, once we hit that threshold at -55, all of those sodium ion channels, the voltage gated channels, are going to open wide open. What does that do? It allows a rush of sodium into the cell. What does that do to the voltage? It's going to depolarize that neuron. The voltage is going to increase radically because we have an influx of all of the sodium ions. What eventually happens is we get to 30 millivolts, and those sodium voltage gated channels are going to become inactivated, and now the potassium channels are going to open wide open. What's going to happen? We have a rush of that potassium outside of the neuron. It takes a while for those to eventually close, and so we then have an undershoot. And what's happened is at this point the voltage has gotten really, really low, but now that permeability, again, those leak channels are going to reestablish that gradient again to negative 70 millivolts, so we could have another action potential. And so if we look at what that looks like in slow motion, we've got a quick opening of the sodium voltage gated channels. You can see an influx of sodium from there and adjacent. That's followed quickly by an opening of the potassium channels. We then have that outflow of that potassium, and then you can see that permeability is going to reestablish us at that resting potential. We could look at what this looks like as we graph the potential over time. Open of sodium, we see a rapid depolarization, opening of the potassium, a repolarization, and then we have that undershoot. And during that time, until we reestablish that gradient at resting potential, we can't have another action potential. Now, so far, we've only been looking at one cross-section of the neuron itself. How does that message move all the way down the axon? Well, if we look at a simulation, it looks like this. Let me slow that down. What really is going on is we're having depolarization of a segment of the axon, which triggers depolarization of the next segment and the next segment and the next segment. So let's zoom in and look at those voltage-gated channels and see what's going on. So if we look at, this is near the axon hillock at the beginning of the axon itself.
So this is where how I was talking about a chain. It's a chain reaction of action potentials, and you can see it works like a wave. This difference in energy or the influx of certain ions will push onto the next section.
We've got some voltage-gated sodium and potassium channels. And so when we hit that threshold, those sodium channels are going to open wide open. We have an influx of sodium ions here, and what they're going to do is they're going to move away from each other since they are going to repel like charges, and they're going to change the voltage in this next segment. What does that do? It hits threshold again and opens up the next sodium channels. Now, why isn't it going to move to the left? Because to the left, we have that repolarization of that segment to the left. So we're opening up those potassium channels, and we have an outflow of those charges. We're not going to be near that threshold for the sodium voltage-gated channels. And so what we're really having on the leading edge of this is a positive feedback loop. We have more sodium coming in, which triggers more opening of those sodium channels. And behind that, we have the undershoot of as we reestablish that gradient again. And so it's going to travel all the way down the axon. Now, this is a simulation of what that electrochemical signal looks like as it moves down. But we have some really long axons in our body. We have an axon that goes all the way from the base of our spinal cord down to our foot. And so it's not going to travel fast enough. And so in a lot of those neurons, they're myelinated. And what that means is they're wrapped with myelin, which insulates that nerve fiber. And so if we look at it like this, what's really going on is we're putting those voltage-gated channels in between that myelin sheath. And I'm not going to get into the specifics of what's going on in here, but if we look at this simulation right here, what it does is it allows that electrochemical gradient, you can compare the two, to travel more readily as it moves farther down the neuron. Now, action potentials succumb to what's called the all or none law. And what that means is that an action potential will occur or will not, and it doesn't depend on how much of the stimulus it gets. It depends on if you hit that threshold or not. And so once we hit that threshold, we can move towards it, we can move away from that voltage, but once we hit negative 55 millivolts, we're going to have an action potential, and that action potential is going to look exactly the same. Now, in this diagram right here, we have the three different potentials that I'm talking about in this video. At the beginning, we have the resting potential. Remember, that's contributed to by the permeability of the membrane to different ions. We then have the action potential, that rapid depolarization and repolarization of the neuron. But what's going on in here, we have graded potentials. In other words, the potential is moving towards or away from that threshold. And so if we think about what's going on there, to really understand that, we're getting information from other nerves. There are synapses here, and those synapses are releasing neurotransmitters. Those neurotransmitters are moving across the synapse, and they're opening up a new form of channel that's important when we talk about graded potentials. Those are chemically graded channels. And so these neurotransmitters are going to dock with proteins on the postsynaptic side of those neurons, and what they'll do is they'll open it up. What does that do? Well, it depends on what type of neurotransmitter it is and what type of receptor it is. There are some that are going to be excitatory. And if we look back at our graded potentials, excitatory means that they're moving us towards the threshold, towards that all or none action potential to occur. What's the easiest way to do that? If we open up the channel and we have a channel that allows sodium to run in, that's going to depolarize us and it's going to move us to that threshold, just like those sodium-gated channels are going to do. Now, we also have inhibitory neurotransmitters and receptors. What are they going to do? They're going to move away from that threshold. What's the easiest way to do that is to have a receptor that's going to allow potassium to rush out, or you could allow chloride to come into that neuron itself, and it's going to push us away from that threshold. So we can think of a neuron as an analog portion and a digital portion. So since it's all or none down here, we're either going to have the action potential or not. So it's like a one.
So this part, we can skip ahead, but really all of this is saying is all or nothing, so it has to reach that threshold, and once it does hit that threshold, it will complete the full action potential, and the various pumps along the way will repolarize and help recorrect the potential. Okay, and this analog digital section is talking about. I'll mute Mr. Anderson here, but what he's saying is that it's intaking all this different information, and so it's getting each dendrite is voting, so to speak. Like, yes, we should fire. This one says, no, we don't have enough potential to fire. And this one has enough potential. This one has enough potential. And it's almost like they're voting, or voting on the process of whether to move forward with the action potential. I'll let him describe it as well.
Summation of all of those signals that's telling that neuron to either fire an action potential if we hit negative 55 or not. And so that is the all or none law. And so in summary, what is the resting potential? It's negative 70 millivolts. It's where a neuron is sitting. It has a potential to do work. It's established by the permeability of the membranes and the sodium-potassium pump. We then moved on to the graded potential, and that's based on all the neurotransmitters that we're getting from connected neurons. And then if we hit that negative 55, we're going to have an opening of those sodium ion channels, and we're going to have an all or none action potential that runs down to the neuron. So that's the action potential, and I hope that was helpful.
All right. So this is that basic diagram of the action potential. And some key things to remember is that if it's resting, right? Think of the visual: red light, yellow light, green light. Rest is red. R and R goes together, makes sense. And the action potential, whether it goes green action, depends on the gradient here in the yellow. This is where all that information from the various dendrites come into play, and whether it reaches this threshold determines if the full action potential actually occurs. So it could be, oh, we have some, oh, we don't have enough, oh, and then once they hit that negative 55, like Mr. Anderson said, every action potential looks the same. And how they communicate with each other, right? How he said synapse. So if a neuron sends information down to its terminal ends, how that gets from the terminal end to the dendrite of another neuron, that is a synapse. Okay. So I'll review what we've gone over so far. We talked about the overview of the nervous system. It's the command center. The basic unit is the neuron. We talked about the structure with the dendrites on one end, the axon, and then we have the terminal ends, the myelin sheath covers the axon, dendrites. Information comes in via the dendrites, travels along the axon from action potential to action potential. And then we have the terminal ends. And this is where that information is transferred to another neuron. Salty banana, remember that helps us remember which side has the most concentration of a certain material, right? So we have the sodium on the outside, and it can travel via those channels, those pumps. And the potassium from the inside the membrane will travel outwards. So we have that depolarization, which is rapid, and all that sodium comes in. And then we have that repolarization, that undershoot, and then back to the resting potential. And think about batteries and charge differentials, the electrochemical gradient, and how the action potentials act in a chain, right? And each one induces the next influx of sodium and potassium, okay? And now our last section is about the synapse, the space in between the neurons, how the information is sent between these neuron cells. And two terms we gotta remember here is excitatory and inhibitory. These will bring the neuron closer to the action potential, whereas inhibitory means it will stop, it will bring it away from the action potential. So excitatory will excite. Think of things increasing. The voltage, the potential is increasing. Inhibit is decreasing, trying to stop something from continuing. And we'll check out that video right now. There we go.
Learn math and science on Brilliant.org.
Brilliant is the best.
Well, after the ad.
Hi, it's Paul Anderson, and in this video I'm going to talk about the synapse, which is a connection between adjacent neurons. That's a pretty strict definition. Remember, neurons can also be connected to effector cells in our body. But if we look at this neuron here and this neuron, the information has to get from this axon to this cell body or to this dendrite, and then the same thing down here. And we use synapses to do that. We're mostly going to be talking about chemical synapses in this video. In a chemical synapse, you have an action potential move down an axon. It triggers the release of neurotransmitters into this gap or this cleft right here. They're going to dock with chemically gated channels on the other side, and that can lead to an action potential on the other side. Some of the early work done with neurotransmitters was done by Otto Lowy. He had this dream of a wonderful experiment in the middle of the night dealing with the heart of a frog. He wrote it down, but when he woke up in the morning, he couldn't read his handwriting. Thankfully, he had that same dream again, and this is how the experiment that led to a Nobel Prize win. He had the heart of a frog that was beating, and then he would stimulate the vagus nerve on the surface of the heart, and what that does is slow the beating of the heart. So we're sending a message to the heart. He then took another heart from a different frog, but he removed the vagus nerve, so there's no neuron there. He got it beating, but he moved some of the liquid from the stimulated heart to the non-stimulated heart, and it slowed down as well. What did that mean? There was something in the liquid. He discovered these neurotransmitters, and he discovered a chemical synapse. In a chemical synapse, a action potential moves down a nerve, down a neuron. It triggers the release of neurotransmitters into the gap that are going to dock with receptors on the other side. Now that's only one type of synapse. We also have electrical synapses, and in electrical synapse, an action potential is going to move down. And again, that's triggered by these voltage-gated channels. Once it gets to that synapse, it's going to open up more voltage-gated channels, and the message is going to go. In electrical synapse, if you have an action potential in the neuron before the synapse, you're going to have an action potential in the one after. What's great about electrical synapses is that they're very, very fast. What's bad about them is that there's no control. So let's talk about chemical synapses. To identify the different parts of it. So this would be where the information is coming in. So on this neuron, and this would be the cell body of the adjacent neuron. So we have a presynaptic side and a postsynaptic neuron. And then this little gap in the middle is going to be synaptic cleft. Now there are structures that hold this in place. It's not just floating there. On the inside of that terminal bud, we have synaptic vesicles, and those are filled with neurotransmitters. Those are different chemicals, chemical messages that can be sent across that synaptic cleft. There's some other anatomy that we should cover. There are going to be voltage-gated channels on the terminal bud. Those are going to be calcium ion channels. We also have docking proteins for vesicles on the presynaptic side, and then we're going to have receptors on the postsynaptic side. And so if we remove all of those terms, let's see what happens. So first thing that happens is we have an action potential that's moving down this neuron here. If you don't understand what an action potential is, I'll put a link to a video that I made on those. So the action potential comes down, and what that's going to do is the depolarization of that neuron is going to open up these calcium voltage-gated channels. Now the electrochemical gradient for those calcium ions is into the neuron itself, and so they're going to move in, and they'll dock with chemicals on the surface of those vesicles. They're going to activate proteins on those surfaces, and what allows those to do is to connect with other docking proteins on the surface of the presynaptic side, and allows those vesicle membranes to merge. They then dump their neurotransmitters out into the gap where they can dock with these chemically gated channels on the other side. So they're sending a message through those neurotransmitters. Now the neurotransmitters will quickly then break apart. Some are broken down, some are recycled back into those synaptic vesicles. But let's see what's going on. So there are a number of different channels in every neuron. They're going to be leak channels for sodium and potassium. What those do is establish this resting potential. We also have voltage gated channels. Those are important in the transmission of that action potential. But here we're talking about receptors. These are chemically gated channels. So when the neurotransmitters gap with it, they can open it up. What does that do? Well, it can move us towards or away from an action potential. So an action potential is when we hit this threshold of negative 55 millivolts. That's determined by these voltage gated channels. But what can happen in an excitatory neurotransmitter, and a neurotransmitter that's pushing us towards the threshold, is that it is increasing the voltage. It's moving it closer to that threshold. What's a quick way to do that? If we have a receptor on the postsynaptic side that allows the flow of sodium into the neuron. That sodium is quickly going to depolarize the neuron and move us closer to the action potential. Now, some of those neurotransmitters may be inhibitory. What they're doing is they're allowing the flow of ions, but these are ions that are moving us away from that action potential. What's a quick way to do that? We could release a bunch of potassium or allow chloride to come into the cell itself. But if you think about it, a neuron is then receiving information from all the neurons that are connected to it that are telling it to either fire an action potential or not. What are they really doing? They're sending either excitatory, those are depolarizing messages, the influx of sodium that is starting to trigger an action potential, or inhibitory. Those are ones that are allowing potassium out or chloride in, and they're stopping that message from being sent. And so we really have an analog side to a neuron and a digital side. The summation of all of those signals, all those neurotransmitters, either tell that action potential to fire or not, and it's going to move down that axon. Now remember, when we get to the end, there are going to be terminal buds here. Again, calcium gates are going to open up, and we're going to have those new neurotransmitters that are sent across. Now, when you remember things, so if you can remember things early from this video, a lot of that has to do with connections in neural pathways in your brain. And what we're starting to find is that when you really remember something, that as we fire action potentials, as we have more of these synapses being activated, what we have is long-term potentiation. And so if we look at what's going on here, if we keep firing this neuron, we've done this in the lab, we keep firing and releasing neurotransmitters. On the postsynaptic side, the cell will actually develop and build more of these receptors, so it's more susceptible to that. And so we're building pathways of memory over time, and that's really what memory is. It's a set of neural pathways that are firing inside your brain. So that's the chemical synapse, and if you can remember a lot of the parts on this pre- and postsynaptic side, you're well on your way to improving your memory, and I hope that was helpful.
Okay, so that was the overview of the nervous system, the neuron, the basic component of the nervous system, the action potential, how that information is transmitted, right? And the synapse, how that information is transmitted from neuron to neuron. And we can go back over the nervous system function lesson just to cover anything we might have not covered in those videos. And I think the most important thing to mention in this part, it talks about the endocrine system here, how it transmits messages through the release of hormones, and that is a slower process, but the effects are longer lasting. Whereas the nervous system, that is the other communication system, and the nervous system uses those electrical impulses, whereas endocrine system uses hormones. The nervous system is quicker, but the effects are not as long lasting as those hormonal functions. We have the neurons, and then the neuroglia, which are the accessory cells, and those work to support the neurons. Here we talked about the structure, and we covered that in those videos. The cell body, the axon. And we have the sensory neurons that are intaking information. Motor neurons, those are the response to the stimulus from the sensory neurons. And then interneurons. We did not cover the interneurons necessarily, but we have the sensory motor neurons, and then those interneurons are only along the central nervous system, and they convey messages within that central nervous system. They can be long or short. Their axons can be long or short. And here it gets into the neuroglia cells that produce a lipid protein substance that covers the axon. That myelin sheath is composed of this substance. And this is a result of that, the work of the neuroglia. Here we have the myelin sheath. That is the protection for the axon, and it helps travel and helps move that electrical impulse more quickly. And there are gaps in between the sheath, and those are called nodes of Ranvier. And when you get a lot of neuron fibers collected together, we have a nerve. And the entire system is separated into the central nervous system, sometimes called the CNS, or the peripheral nervous system, the PNS. And the central nervous system is central to your body. It's made of the brain and your spinal cord. The peripheral nervous system, those include the cranial nerves and spinal nerves. Cranial originates from the brain, while your spinal nerves originate from the spinal cord. And we can divide that into the somatic and autonomic nervous systems. And those will be a topic for the next lesson. And we'll get more into that next time. But thank you all, and let me know if I can help you with this lesson further. Email me, message me in Canvas. You know how to reach me. But good luck. Thank you, and goodbye. See you next time.